Good afternoon, respected members of Tatweer platforms and universities, colleagues, students, and friends. Welcome to you all to a new international webinar organized by Tatweer platform. I'm Dr. Ahmed Al-Amar, and I'm the director of Tatweer platform, and I'm also a lecturer at the College of Medicine, University of Babylon, Iraq. I will be moderating this session, and we will have the registration and certificate link will be sent to you at the end of the webinar through the chat box. And also we will have a question session at the end of the webinar. So if you have any question, please write it down, and then you can write it in the chat box at the end of the webinar, and we will try to answer it. Also, if you have any technical problem regarding the voice or the screen, or if you have any notes, you can write it in the chat box during the webinar, and I will have a look on them and will try to answer them. Let me welcome our eminent invited speaker, Dr. Pallav Sengupta, who is a senior lecturer at the Faculty of Medicine at MAHSA University, Malaysia. And he is going to talk today about COVID-19 and male infertility. We are very grateful for Dr. Sengupta for his valuable time and for accepting our invitation and for sharing his rich knowledge and research experience with us.
Thank you, Dr.
Before I read the short biography of Dr. Sengupta, I would like to say that he is a friend and a great collaborator, and I have approached him 2 years ago, and I asked if there's a possibility of collaboration because we shared common research in andrology and specifically in the oxidative stress and sperm DNA damage effect on male infertility. Dr. Sengupta, without hesitation, he agreed for collaboration, and since then we had a great collaboration together with him and with Dr. Duta, the same department in MAHSA University, and this collaboration has been very fruitful, and we managed to conduct many original researches and publish many original articles and reviews in high impact journals. So thank you very much, Dr. Pallav, for your collaboration.
Thank you, Dr.
Now I'm going to, thank you so much. Now I'm going just to provide a short biography for Dr. Sengupta. So Dr. Pallav Sengupta is a senior lecturer at the Department of Physiology, Faculty of Medicine, Bioscience and Nursing at MAHSA University, Malaysia. Dr. Sengupta has published more than 100 research articles and several book chapters with current Scopus H index of 21. He has worked as the head of physiology unit and assistant professor in the Faculty of Medicine, Dinkel University College, Malaysia. Dr. Sengupta has pursued a Bachelor of Science honors and Master's in Physiology and PhD in Reproductive Endocrinology from the University of Calcutta, India. During his PhD, he was awarded the Young Scientist Award by the Physiological Society of India in 2012. He had started his teaching career as a lecturer in the Department of Physiology, Vidyasagar College for Women, University of Calcutta, till date, Dr. Sengupta has guided 14 research projects for Master's Medical Sciences and the Bachelor Degree of Medicine program, the MBBS. He is currently working with 4 research grants, including Fundamental Research Grant Scheme, research grant from the Ministry of Higher Education, Malaysia. He has received the highest research publication award of 2019 from MAHSA University, Malaysia. Dr. Sengupta has also pursued research internship in andrology from the American Center for Reproductive Medicine, ACRM, Cleveland Clinic, USA, which is a famous one. I know it's a very famous one. And presently, he is a research collaborator and international research mentor in the IRCM Cleveland Clinic, USA. So this is just a short biography for the rich biography of Dr. Sengupta, and we are very thankful and very grateful for his collaboration and accepting our invitation. So a very warm welcome to Dr. Sengupta. Now I invite Dr. Sengupta to share his screen and present his talk. Dr. Sengupta, please.
Yeah. Thank you, Dr. Ahmed. Good afternoon and good evening to everyone, wherever you are. First of all, I want to thank Dr. Ahmed for the nice introduction. And as he has mentioned, that we are working for several years, and we have come up with several good quality articles, several good quality research, and we are still continuing it. I express my sincere thanks to him. And I also thank the organizing committee for this particular scientific program. I also thank the University of Babylon for inviting me for this particular webinar. So as Dr. Ahmed was mentioning, that today I'm going to talk on the effects of COVID-19 and male fertility. So, and its evidences and hypothesis. So the evidences, what we have got till that, and the hypothesis, those have been already put forth. We will discuss on that. And we all know that still the pandemic is continuing, and several aspects of this particular topic is still not revealed. It's not unveiled. So whatever available, whatever information we know, we will discuss on that. So before I start, I'd like to confirm, is my screen visible to everyone, Dr. Ahmed? Dr. Ahmed, my screen.
Dr. Sengupta, it's not showing. I will send you another request for sharing so that we can have your screen.
Okay. Okay, I think I can try.
Yeah, now it's showing now, Dr. Sengupta.
Okay. Okay. Thank you, Dr. Ahmed. Okay, let's begin with today's topic, COVID-19 and male fertility. As I was mentioning, we will discuss about the evidences that we have got and the hypotheses, those have been already put forth. So, as we all know that since last 2 decades, we all are experiencing several pandemics, and related to coronaviruses, this is our third experience, COVID-19 and the infection by SARS-CoV-2. It started in 2003 with SARS-CoV infection. And in 2013, we remember we experienced the infection of MERS-CoV. So this is a new experience, what started in December 2019 with a new coronavirus. And previously, when the first cases were coming, that time everybody was not very sure regarding. They were not very sure regarding the infectivity, the mode of infectivity, the fatality of this particular viral infection. But day after day, when the reports started to come, many things got revealed, and now we have several information on this, not only related to its infection and pneumonia. We have information; those are based on several other systems. So today we will focus on the infection, the mode of infection of this particular virus, how it affects a male reproductive system, how it causes disruption of male reproductive functions. So this is a coronavirus, as I told you, and the global impact, it started in December 2019. This is SARS-CoV-2, severe acute respiratory coronavirus 2, and it causes the disease that is known as COVID-19, coronavirus disease 19. This is a beta coronavirus. It is a mutated version of SARS-CoV. Several reports, they have already published it. And in December 2019, the first detection was done in Wuhan city in China, and after that it started spreading like a forest fire. And in 11th March 2020, World Health Organization, they declared this particular disease as pandemic. Because by that time it had spread over several countries, several continents, and it had affected millions of people, and several fatalities had happened. So in March 2020, WHO declared it as a pandemic. So we will start with the basic structure of this particular virus. As we know, it is an RNA virus, and it is having several spike proteins around it. And this spike proteins, this S proteins, the basic genetic studies, they have showed it is a mutated version of the SARS-CoV, and the origin, mode of infection, everything is quite similar to that of SARS-CoV infection. And as it is an RNA virus, it is having an envelope, a nucleocapsid protein, and those are quite similar with the other RNA viruses. So this is SARS-CoV-2, and this coating protein, these spike proteins, these are having variable receptor binding domains. Like SARS-CoV, like MERS-CoV, it is also having several variable receptor binding domains, RBDs. And proteolytic analysis of these proteins have showed that it is a mutated version. And that's why it is showing several differences with SARS-CoV and MERS-CoV. With some similarities as well, it is showing several differences with the previously reported coronaviruses. These RBDs, what they do, these receptor binding domains, they bind with one specific receptor that is known as angiotensin converting enzyme receptor type 2, that is present all over our body, in our lung, in our heart, in our kidneys, in our GIT, in our reproductive tract, and it facilitates several functions, not only related to male reproduction. We all know that. And the presence of this particular receptor actually makes us more prevalent. It makes us more susceptible to this particular infection. RBD of SARS-CoV-2, it binds with the ACE2, and it facilitates its activation, its invasion to the affected cell. Okay, so this is the key protein which is actually now reported in several publications. And its affinity, the affinity of the RBDs towards the SCE2, it is also a determinant of the infectivity. If the cell is having several SCE2 over it, the infectivity will be higher. And from the analytical studies which have confirmed that this particular virus, by analyzing the RBDs, their nature, it has showed that this particular virus is actually came from bats. The origin is also confirmed by analyzing the nature of the RBDs. Here I have put that RBD of SARS-CoV-2 appears to be mutated version, which is closely related to the virus RATG13, sample from bats. So it has mutated from that particular protein, and that's why it is now evident. The source is now we know that it is from bat, and after mutating was able to infect other animals also. This is quite interesting. The comparison between SARS-CoV and SARS-CoV-2, if we compare it, we can see the infectivity of this particular virus is more towards human. The human-to-human transmission is more than what we have seen in SARS-CoV. Why so? Because the affinity of this particular viral proteins are more for humans. And it has also been seen that for rodents, the affinity of this particular protein has decreased. So rodents are not that much affected. But some other animals like ferrets, pangolins, in those animals also we can see the affinity is quite higher. And this is the thing which is, makes that particular animal, that particular human being more susceptible to SARS-CoV-2 infection. So sometimes the pangolins or ferrets, they act as the intermediate host from bat to the pangolin to human being. So they serve as the intermediate host of this particular virus. So as I was telling, the diagram is also showing the primary host for this particular infection is bat. And after that, the thing is, we have determined that through this analytical studies that pangolins and other animals could be served as the intermediate, and the human-to-human transmission has also increased. So spike proteins, how they actually bind with this SCE2, and how the other proteins are helping in the invasion of this particular viral components in the cells. So here I have put the cellular entry, how it enters the affected cell. So spike proteins on the surface of the coronavirus binding to the SCE2 receptors, they target it. The spike proteins, they target the SCE2, those are present over the cell that is going to be affected by the SARS-CoV-2. And what happens after binding to the SCE2, when the RBDs bind with the SCE2, there is another protein which is also similarly important, that is transmembrane protein, one kind of protease, certain protease type 2, that cleaves this entire structure, and it helps in the activation and in the invasion of the viral components. So cleaved SCE2 and activated spike proteins facilitate viral entry. And SCE2 receptors are highly expressed in several cells, in our lung cells, in our cardiac muscle cells, in our testicular cells, several testicular cells. It has been found in our Leydig cells, our Sertoli cells, our germ cells. And from that particular context, the idea that that concept of its infectivity in male reproductive organs, male reproductive tissue stem. And later on, it has been reported that he is disturbing several male reproductive functions. So the reports started coming regarding its effect on several aspects of male reproductive function. And there is another aspect, that this particular cleaving enzyme, TMPRSS2, this protease, its activation is androgen dependent. So this is the other way which shows that the activation or invasion of this particular virus or viral components is somehow dependent on the male reproductive hormones, and the expression of ACE2 in several male reproductive tissues also confirm that particular aspect. So this is the mode by which actually this particular virus actually affects the affected human being. Here I have put one side, the left side is for mild infection and right side is for the severe infection. This particular diagram is published in Lancet by Lee et al. This year. So here you can see that when the virus comes in, and if the viral load is not that much, if it is not severely affecting that individual, it actually it actually activates both innate and our adaptive immune system. And the resident macrophages, they are efficient enough, they are enough to fight against it, and some parts of the adaptive immune system, affected T cells, they also fight against it to remove the viral load. And as a result, what we can see the person is either mildly affected or not at all affected. So not at all affected means it is either maybe the person is affected with very low viral load and is not showing any kind of symptoms. Okay, but where, and also in the circulation you can find very less amounts of affected T cells or naive T cells, which will indicate that the infection is not that severe. But what happens in case of severe infection? In case of severe infection where the viral load is too much, that time, along with all these T cells, we can see several other adaptive immune cells are coming, and what happens, the integrity of the epithelial cells, it also gets disrupted, and SARS-CoV-2 start attacking the lung capillary endothelial cells, which leads to the infection of this particular virus. Alveolar macrophages or epithelial cells could produce various pro-inflammatory cytokines and chemokines. Upon this change, monocytes and neutrophils were then chemotactic to the infection site to clear the exudates with the virus particles and infected cells, and resulting in uncontrolled infection. Here you can see that several components are released, several pro-inflammatory cytokines are released in the circulation due to the excessive viral load. And other than T cells, we are also taking the help of neutrophils and other cells, which are causing the ultimate effect. Meanwhile, the direct actor on other organs by disseminated SARS-CoV-2, the immune pathogenesis caused by the systematic cytokine storm, which several publications, several reports are reporting, the cytokine storm and the microcircular dysfunction, it actually ultimately causes all these symptoms, and the systemic effects and specific system-based effects can also be seen, also be also can be recorded here. Okay, and in that particular patient, you can record the symptoms of severe infection. So it is clear to all of us that it infects the human system, its respiratory system, its other system, but does it affect male fertility? And if it is so, how it affects male infertility, how it affects male fertility, and why male particularly? So several reports, they have already showed, and I was also mentioning that in male reproductive tissues we can see several expression. We can see the expression of ACE2 is quite evident in Leydig cells, that is concerned with our steroidogenesis, Sertoli cell concerned with our spermatogenesis, in different of our germ cells as well. It is already expressed. And the other fact that I was mentioning before, that the expression of the cleaving enzyme, the serine protease, that is also androgen dependent. So the incidence of COVID-19 among men is reportedly higher. And here you can see the ratio of occurrence in male to female is about 2.7:1. And the fatality, the ratio of fatality of male to female, it is 1.6 to 2.8. So there are several factors which actually make males more susceptible to this particular infection than female. And there are a few studies also, evidence-based literature, and which actually presented the possible association of the male fertility, its disruption, with SARS-CoV-2 infection. Later we will discuss all this. So men with COVID-19 showed substantial injury in the seminiferous tubule and reduction in the Leydig cell count, Leydig cell number. Okay, and only in one case, this is we have to confirm that yes, we are recording several changes, several male reproductive changes due to this particular infection. But the occurrence, the presence of the virus particle inside the male reproductive tract is only evident from a single study. The other studies, they are not directly reporting the presence of this particular virus inside the male reproductive tissue, but the effects are evident. How the effects are coming? There are several hypotheses already presented, several reports, they are already assuming several aspects of this infection and infection mediated effects to the male fertility. We will discuss on that. But in only single case, in one case only, we have seen the presence of the virus particle in the testes. And testicular autopsies of 10 diseased men, male COVID-19 patients, showed the presence of this particular component. So here I have put some of the reports which have showed direct relation of the particular this particular virus infection and their correlation with semen parameters and some other things of male reproductive functions. So I'm not going to discuss all these things in detail, but here you can see most of the studies in their findings, they are showing that the SARS-CoV-2 and its detection is negative. But as I mentioned, in only one study, I think probably, if I'm not wrong, it's by Lee et al., which have showed that positive occurrence of this particular virus in our male reproductive tissues. And in this aspect, another study is also important by Ma et al. This year, which first presented the endocrine changes, the hormonal changes which are directing towards the male reproductive disruption. It has first reported that a reduction in testosterone is there, LH elevation is causing, and that showed another direction by which this particular virus may work on our male reproductive tissues and can disrupt its functions. So these are some of the reports which we have already seen in several scientific publications. Those are not indicating the presence, the positive occurrence of this particular virus in the semen or in the testes, but they are reporting changes in the semen parameters. Here you can see Seger et al. They have reported reduced sperm concentration and motility. Some other reports, they are also reporting reduced sperm viability, because the sperm viability, the regulation of the apoptotic factors, which are also quite dependent on the angiotensin system. So if that particular system is not regulated, if that one is disrupted, definitely the semen parameters will be disrupted as well. So most importantly, a publication by Lee et al. In Lancet, it is reporting that it is a very interesting and important study that is showing that the effect of this particular viral infection on male reproductive functions. The group, this particular research group, they have conducted in 23 recovering patients, 23 controls, and I think 6 or 7 diseased patients. And in them, they had actually conducted the studies, and they have reported autopsied testicular and epididymal samples showed epididymitis and testicular interstitial edema, high deposition of inflammatory markers, in the testis, exudation of the erythrocytes in the testis, seminiferous tubule, thinning and increased number of seminiferous apoptotic cells. So they have actually, first time they have showed that these are the changes which are directly occurring to the epididymis, to the seminiferous tubule, to the testicular tissues. Previously, during the SARS-CoV infection, several studies were reported which showed the presence of orchitis, epididymitis. Several things actually have reported. But during this particular pandemic, several hypotheses had come out which were showing that orchitis-mediated oxidative stress, and several things may happen which may ultimately disrupt male reproductive functions, spermatogenesis. But the thing is, this particular study showed that evidence that yes, it is happening, and that's why the spermatogenesis is disrupting, the semen parameters are altering. In this particular study, men who were in recovery phase, this is also important, recovery phase of COVID-19, among them 39.1% of population have been found as oligospermic. After the infection, when they were recovering, and that time it was found that after infection, 39.1% of the population, they had become oligospermic, and 60.9% were presented with significantly higher number of seminal leukocytes. So what it indicates towards oxidative stress, it indicates towards leukocytospermia mediated probably several other studies, and the future studies, they may confirm it, leukocytospermia or other things. But it directly indicates towards the oxidative stress mediated male reproductive disruption. So we need this kind of studies, and emerging evidences will point out towards the SARS-CoV-2 infection-mediated testicular disruptions. And we need the large-scale studies, though it's difficult to conduct. We all know that. which are required for better understanding of the testicular viral dynamics. Till date, we do not know that the virus comes inside, it goes out or no, or only the viral particles mediate the effect. But we believe that in future studies, we will also be able to see the proper viral dynamics. The condition, the study, what I have mentioned before, a single study where we had seen the presence of viral particle inside the testis, that one was with huge viral load. So that's why people may say that due to the excessive viral load, it has also infected testicular cells in that extent. But later on, we believe that the studies will show the proper viral dynamics and will elucidate the mechanism how it works. This one, that the oxidant-sensitive pathway of SARS-CoV-2 mediated function, everything we have actually, this hypothesis we have previously reported in one of our publications that is published in Reproductive Sciences this year. We have shown that how this particular viral infection actually causes male reproductive disruption. Though we are already only focusing on the semen parameters, testicular function, there are several ways by which this particular virus can affect male reproductive functions, male fertility. As I told you before, that this particular virus, it binds with SCA2, TMPRSS2, this particular protease. Its activation is dependent on androgens. All these things are there. But several other comorbidities, those are also very important. We should take care, we should consider all those things together, which can also contribute to male reproductive disruption. So here in this particular publication, what we have showed that SARS-CoV-2 activates oxidant-sensitive pathways for generating the oxidative stress. And this is basically the NF-kappa beta pathway through the binding of toll-like receptor type 4. Though the report that is directly indicating the oxidative stress, its generation in the male reproductive tissues through this particular virus is not evident, but there are reports which are showing the oxidative stress generation by this particular virus, the pathways, and how they stimulate the release of cytokines, and they are causing the inflammatory responses. All these are quite evident. So in this particular diagram, what we intended to show that after invasion, after the infection is occurring, this infection causes a cytokine storm, and this cytokine storm resulted in infection, and this inflammation. This inflammation, it can affect epididymis to cause orchitis, and it can cause oxidative stress. This particular inflammation, the release of huge cytokines, it can cause oxidative stress, and oxidative stress, it can affect several of our cellular functions. It can damage Leydig cells, it can damage our Sertoli cells, it can damage our spermatozoa, and can reduce sperm cell count, can disrupt spermatogenesis, can disrupt steroidogenesis. So these are the direct effects in the reproductive tissues. And through indirect effects, through the comorbidities, through the psychological stress, it can also affect male reproductive function. And it has also been reported in some publications, you can see later on, that some of the antiviral drugs like ribavirin, it has been reported to cause sperm DNA fragmentation, oxidative stress, and so on. So all these factors, they work together in the affected patient to disrupt the fertility status of that individual. This is another diagram from another hypothesis, another theory that we have already reported in another publication in Contraceptive Reproductive Healthcare this year. That how this particular virus, how this particular disease may be associated with sperm DNA fragmentation. Surprisingly, this particular area is not that much covered, is not that much explored. And we are still waiting for the reports to come with actual evidences of the sperm DNA fragmentation mediated by that infection. So here what we had proposed, similar to our previous diagram what I have showed, that SARS-CoV-2 infection actually it mediates oxidative stress. And this oxidative stress can directly affect sperm DNA structure, chromatin integrity, and can cause sperm DNA fragmentation. I was also mentioning about the antiviral drugs like ribavirin, which is also used for the management of COVID-19, but has been reported to cause oxidative stress and sperm DNA fragmentation. Along with all these factors, comorbidities, age of that particular male patient, everything can contribute to cause the sperm DNA fragmentation, sperm DNA damage. So, though this is not presented, though this is not reported in an original report, it can be understood, it can be assumed that how this particular virus or its effects, the consequences can affect male reproductive function, can affect sperm DNA integrity. This is another diagram from our another report that we have published in Human Fertility recently that shows that the TMPRSS expression is androgen dependent. Here you can see we have shown that both ACE2 and androgen receptor, their loci are present in X chromosome, and when they are activated, ACE2 expression will be there, and when that testosterone level is high, when the androgen receptor expression is high, it can facilitate the expression of the proteolytic enzymes, TMPRSS2, the protease. So this is another aspect, as I was mentioning before as well, that the binding proteins, the activated proteins, they are also androgen dependent. We have reported this one in one of our articles. And here, similar to our other articles, we have again showed the oxidative stress, but the important things what we have put forth in this particular publication is that after following the oxidative stress, what we can assume that definitely the combating mechanism will come and will try to mitigate the oxidative stress. The HPA axis, hypothalamo-pituitary adrenal axis, will get activated. More ACTH will be formed, more glucocorticoid will come out and will mitigate the oxidative stress. But the thing is, reports are showing that this virus is, when it is infecting the cell, it is also releasing some viral amino acids which can mimic ACTH. So as it is mimicking ACTH, actual ACTH cannot go and bind to the adrenal gland and cannot produce enough glucocorticoid. So glucocorticoid level will be very less. So as the glucocorticoid level is very less, it is not able to combat oxidative stress. So we are generating oxidative stress, but our endogenous mechanism, the mitigating system, is not working properly. So the oxidative stress mediated disruption will continue. And on the other way, this particular glucocorticoid, what it actually causes, this glucocorticoid, it actually crosstalks with the HPG axis, hypothalamo-pituitary gonadal axis. When there is a high glucocorticoid level, it crosstalks with HPG to regulate LH level. So as the glucocorticoid level is very low, it cannot control the LH level. So the LH level remain very high, which is already evident. I was telling before, it is already evident in the reports of Ma et al., who actually taken the blood samples from the 81 individuals and they analyzed the androgen level, LH, FSH and everything, and have shown that lower testosterone level, higher levels of LH, FSH and lower testosterone to LH ratio. So in the affected patients, in the recovering patients, what it is observed, the expected things are that we can see hypogonadism, lower levels of testosterone, but this is not due to the lower level of gonadotropins. The gonadotropins are already high. So how it is causing, how it is affecting the testosterone level, how it is decreasing it? The theory is, the hypothesis here it is that as it is not affecting through HPA axis or HPA-HPG crosstalk, it may have direct effect on the Leydig cell. Definitely through the oxidative stress, it may affect our Leydig cell to decrease the testosterone profile. So this is the way how it affects our testosterone profile. And in aged persons, in the persons who are already suffering from other reproductive diseases, they may suffer from testosterone deficiency. Not always, but the thing is that particular reproductive disorder or aging or comorbidities may also boost the oxidative stress further, that may ultimately result in decreased testosterone profile. And the ACE2, which converts angiotensin 1 to 7, this ACE2, as it's being continuously utilized by the viral particles, virus, it is not able to do its normal functions. So as it is not able to do its normal functions, as we all know, it is intricately related with steroidogenic functions. And as it is intricately regulating testosterone production, it cannot perform that function. So in that way also, testosterone level may decrease. So this one we have published in Human Fertility, the intricate mechanism how this viral infection can cause endocrine changes, hypogonadism, and is it related to HPA-HPG axis? Is it related to inflammation or no? The similar thing what we had previously published also in another journal, that is this particular testosterone decrease or the hormonal endocrine disruption is due to the disruption of the endocrine axis? Is it due to the disruption of the testicular adrenal crosstalk, or maybe due to the disruption of the HPG functioning? What we have proposed, as we have seen that gonadotropin levels are already high, and they are not playing in testosterone regulation, but still testosterone level is low. This points out towards the oxidative stress and the way of secondary inflammation. The cytokine storm that is actually affecting the testicular cells, Leydig cell, Sertoli cell, they are actually causing this particular damage. So here we had focused that we can rule out the regulation by testicular adrenal crosstalk. We should focus on the mechanistic point of view. We should focus on the action through the oxidative stress and the cytokine storm. So this one also we have published before. So in conclusion, I'd like to mention that in this particular pandemic, we know it's still continuing. Every day thousands of research are coming out. Several of those are also related to andrology, male reproductive functions. So this particular emerging, this particular condition is actually helping us to understand the viral mechanism, its infection, how it affects a mechanistic way, in better way. Every day, several mechanisms, several hypotheses, several original reports are coming out, which are helping us to understand the actual mechanism through which it may work. Maybe later on with some other reports, we may change our ideas. The mechanism, the concept may change, but till that, what we know through which we can only conclude, we can interpret in that way. So emerging evidences are suggestive of adverse effects of COVID-19 on male reproductive health, both immediate and long terms. So immediate effects, how it is immediately affecting the reproductive tissues and how it is disrupting the semen parameters. In long terms, how it is disrupting the endocrine function and their mediation in reproductive functions. That can also be, further clarified later on by upcoming research reports. Reports pertaining to SARS-CoV-2 infection-mediated male reproductive disruptions has depicted that this virus is potent to cause testicular inflammation, as I told before. Disintegration, as I was focusing on the very important report by Lee et al. Very recently published in the Lancet, disintegration of the seminiferous epithelium, testicular tissues, and so on, and epididymitis as well. It has already been reported that in epididymis it is causing inflammation like the SARS-CoV infection before. It affects epididymis. Increased apoptosis as a dysregulation of SCE2 is there. The SCE2 controlling activity of the apoptotic factors has been withdrawn. So apoptosis of the testicular cells are there. So increased apoptosis of the testicular cells as well as alteration in the reproductive hormonal milieu. So all these aspects till that we have found in several reports, several review articles, several hypotheses. So by this, I think we have understood most of the mechanisms how this particular virus invades male reproductive tissues and causes male reproductive disruption. So that's all. These are the references that I had utilized in this particular talk. And I again want to thank you all the participants for patiently listening to this particular discussion. I again also want to thank Dr. Ahmed for inviting me for this particular webinar, and all the organizing committee members for the same. Thank you.
Thank you very much, Dr. Sengupta, for this informative and up-to-date lecture. And it's really an inspiring lecture. And we know that the COVID-19 disease and its mechanisms are still to be elucidated, and we still don't know many aspects of the exact mechanism of the disease. So thank you so much, Dr. Sengupta.
Thank you, Dr.
Dear all, now we will have, I will send you the link for registration, and then so that you can register. You will have it for 5 minutes, and then after that we will be taking the chat questions. Okay, thank you.